algunas palabras. Bueno, luego nos puedes traducir, darles la bienvenida a todos. No sé si hay alguno de los de familia española que creo que se van a conectar. Y bueno, pues que creemos que es muy buena ocasión para dar a conocer a todas las familias los proyectos en marcha. Y bueno, pues que cualquier cosa, desde luego, estamos a disposición de todos. Great. That was Beatriz Olona, and she is co-founder of the Andres Marcio Foundation, and she would like to welcome everybody. She was wondering if anybody from Spain has joined, and is really excited about the opportunity for us to talk about the various projects that are being co-funded by the two organizations and by projects that are not being funded by either of us, and to provide an update to the community.
Hello.
So, hello.
Gisele Bonne.
Hi, Gisele. How are you? Why don't you go ahead and introduce yourself next?
Yeah. I just need. Okay. Now I have the screen as well.
Great. Fantastic. Gisele, do you want to introduce yourself?
So Gisele Bonne. I'm a researcher at the Myology Institute in Paris, and I'm heading a team working on congenital muscular dystrophy related to LMNA-associated gene.
Great, thanks so much, Gisele. I'm going to have, do you want to introduce yourself?
Sorry?
Salome, can you introduce yourself?
Yeah, hello. I'm Salome. I'm from Iceland. I'm the founder of the Emily Dreyfus Muscular Dystrophy patient Facebook group. And I'm just here to listen.
Great, thank you so much for joining us, Salome. Michael Wood, would you like to introduce yourself next? I think Michael is still on mute. Let me see if I can unmute people. Rachel, can you actually unmute him? Is that possible? Mark, would you like to introduce yourself next? Oh, there's Michael. Michael, is that you?
Sure.
No one is muted, Anne.
Okay, everybody's unmuted?
Yep.
Okay. Is there anybody else who would like to introduce themselves? Dan Lillegrand, I see that you're on as well. Would you like to introduce yourself? Well, I think we're having trouble. Maybe Dan, would you like to introduce yourself?
Saludo al equipo. Comprobación del sonido.
Well, maybe what we'll do is we'll skip the introductions of everybody to one another because it appears to be a little bit problematic. So what we're going to do is I'm just going to introduce our other speaker, Georgia Sarkala Brugada. Dr. Brugada, would you like to just say a couple of words about who you are?
Hello, good evening from Spain. I'm a pediatric cardiologist working in Sant Joan de Déu Hospital in Barcelona, Spain. And yep, I'm so happy to be here and share with you a little bit of what we are doing as well.
Fantastic, great. Thank you so much, Georgia. So what we're going to do is I'm just going to walk us through 5 introductory slides, and then the rest of the hour will be open to discussion and questions from the community. So the point of this LMNA community update is to provide an update to everybody in our LMNA community about the current research and what is currently being done. I hope everybody can see my slides. The agenda for today's discussion is first to just provide a brief update on a fantastic meeting that we had in Barcelona on October 19th and 20th, and this was the 2nd annual LMNA scientific meeting. Then I'm going to touch briefly upon our registry numbers in the Congenital Muscle Disease International Registry, talk just to provide you just with a brief update on some clinical research and some basic science research, and I don't expect that this will take more than about 5 to 10 minutes, and then we'll open up the floor to questions, and maybe we can have Gisele Bonne, Dr. Bonne and Dr. Brugada both provide us with updates on their current research. So this was the second annual meeting that took
and we've lost you.
place in Barcelona. And this meeting, can you hear me? Oops. Can you hear me now?
Yeah, perfectly.
Yeah. So we had 29 participants from all over the world joining us. And this meeting was co-funded by the Andres Marcio Foundation and L-CMD. The purpose of this meeting...
T'estic explicant la diapositiva, que va fer la segona anual de l'LMNA-associated meeting a Barcelona.
The purpose of this meeting was to look at and talk about how the heart is affected in LMNA, what we currently know about LMNA, what we don't know, and what the priorities should be for future research. And the idea behind this meeting is to bring together a group of experts every year in order to reconfirm what the priorities are and to accelerate research in LMNA. So very briefly, I also want to talk about the registry, the Congenital Muscle Disease International Registry. And I'd like to ask all families who are not currently registered to register, to send in a copy of their genetic report if they haven't done so, to complete an annual survey, and to consider transferring any muscle biopsies that are not currently being used for research to the CMD tissue repository. And we are happy to help you in doing that if you have any questions. Here is the total number of registrants in the CMDIR from 71 countries, and the number that everybody on this phone call is interested in is how many LMNA registrants do we have, and we currently have 45 registrants, which is 2% of the entire registry. So our goal is to increase this number to at least 150 by the end of next year. And we are really working actively to ask adults also with LMNA to register, because the majority of people who are registered currently are children who are affected who are registered. And you can see that we have registration from multiple different countries, and these are just the countries where we have the highest numbers of registrants. In terms of clinical research, I just want to share with you that there are two important studies that are currently underway, and Dr. Bonne and Dr. Brugada can answer additional questions that you might have about these studies. Several of you already know about the Retrospective Natural History Study, and this is a study that is taking patient files and patient records from several different countries and is analyzing how things change for children and adults who had the onset of disease under the age of 5 over time. And what we are specifically interested in learning is what age do breathing issues start, at what age do heart issues start, how many children and adults acquire the ability to walk and how many don't, and how does this change depending on what your mutation is. We think this study will be a very important study because currently, if you look in the medical literature, the largest group of LMNA patients that has been published is a total of 11 patients from one center. So this really will contribute greatly to our understanding of how LMNA affects children and young adults. The second study which Dr. Brugada will tell us some more about. And this study, our goal is to enroll 60 to 70 kids and teens between the ages of 1 and 21 by December of next year. It is a 3-year study, and Dr. Brugada will tell us more about what participation involves and perhaps share with us some of the early data that is coming out of this study. In terms of basic science research, L-CMD and the Andres Marcio Foundation have funded Dr. Bonne, who just recently completed a 2-year grant to evaluate a combined gene therapy approach of transplicing and a viral vector. And now in January, she will be starting another 2-year grant to test a strategy using siRNA. This term may be somewhat familiar to some of you in its application for exon skipping in Duchenne. So she's going to use a similar approach combined with AAV or a viral gene therapy to see if that will actually yield a positive result in patient cell lines and in the mouse model. Dr. Howard Warman and Dr. Antoine Mouchir have been working on using protein kinase inhibitors. This is a compound to specifically target the heart issues in the mouse model. And they completed several studies that show a small improvement in survival, but a significant improvement in mouse heart function. And we're currently looking at ways of funding additional studies to specifically look at a protein kinase inhibitor produced by a company called Allomec. And there will be future updates, I'm sure, in the next year on that particular compound. And then finally, a drug screening project has been funded together with a fundraising campaign led by 2 Australian moms called Beat for Life and Cure CMD and the Andres Mastro Foundation. And this is to fund the scientists in San Diego in the United States to create models in a dish of muscle cells and heart cells from patients with LMNA, and to expose these cells to drugs to see if you can actually measure an improvement in how these cells look. And the purpose of this particular study is to identify new disease mechanisms that we can target and identify additional potential targets and therapies that can then be tested in the mouse model. So I'd like to just conclude that the current priorities for the Andres Marcio Fund targeted $250,000 to support ongoing enrollment in the LMNA heart study and to raise $50,000 by the end of next year to support ongoing research projects, to increase registration in the registry, and to increase participation in the LMNA heart study. So I'd like to stop there, and I'd like to open the floor up to anybody who has any questions. And really, I'd like to first say that there are no stupid questions, and any question is fair game. And I open up the floor to you and hope that there will be some interesting discussion that will follow. So does anybody have any questions? Okay, maybe I'll start with the first question then. Dr. Brugada, could you explain to us a little bit more about the LMNA heart study, what your goals are for that study, and why you think this study is so important?
Yep. Is that okay?
Yeah.
Okay, great. So what we are doing with the heart study is that after seeing the evolution of some patients that have a cardiac screening and receive the normal and the basic screens exams for heart checkups like echo, Holter, and ECG. We've seen that most of the patients, all these tests are normal. What we were thinking that when we see that something is abnormal or something is not really how it should be, by those tests we think that we are a little bit too late. So that then it's very difficult to go back to the basal point. So we would like to know, to see if we can find predictors of arrhythmias and important heart problems such as sudden death that we could kind of predict doing other tests, like more advanced tests that we have protocolized, like doing a very special echo, which is a 45 minutes long echo, and then there is a long work of offline analysis. Then we do a test, an electrophysiological test, that it's a way to see how the electrical currents and the electrical cables of the heart work. Okay, this is something done by a vein, like a puncture, a normal puncture of the vein, just for a blood test. Then we go with a little, very small catheter up to the heart, and we see how these cables do perform. And finally, what is the most important part of it, but not the only one, is that we implant a very small device, which is a Holter, a subcutaneous Holter. It's something that it's like a little chip that we put under the skin, and we can record and receive the information for beat to beat of the heart during 3 years, and we receive this information via internet. So there's no need for the patient to move back to our center. So we are performing this since February, March. We've enrolled 28 patients so far. We've performed, we've done completely done 12 patients, and we are going to do 5 patients in the following days. And what we see that we have amazing results of what we can see on the heart, that we can go before and check before the heart and see how the heart is before it gets too bad or too sick. So it's a very promising study, and we are really proud about all the work we are doing and all the work that Fundación Andrés Marcio is doing just to help all these patients to receive, to be part of this study.
Great. Georgia, I don't know, there may be some families from Spain on the phone as well. Did you want to just summarize what you said in Spanish as well?
Yep, I think there is Jesus Marcio, and I see that I think the family from Tarragona, they are both enrolled already. They have gone through the study, but I'm more than happy to share with them. Entonces, yo creo que somos Jesús, Beatriz, estáis aquí, y Rosamari debe ser la mamá de Luis, si no me equivoco. ¿Es así?
Sí.
Ah, vale.
Sí, sí.
No, era el resumen del estudio que ya conocéis de primera mano, casi mejor que yo, donde hacemos no solamente el estudio básico del corazón, sino que hacemos la ecografía avanzada, el estudio electrofisiológico para ver cómo funcionan los cables eléctricos del corazón, y lo que nos da la información a 3 años vista es de la actividad eléctrica del corazón mediante el implante de un dispositivo subcutáneo que nos permite estudiar vía internet cómo está el corazón del niño de una forma directa. No sé si hay otras familias españolas aquí que estén. No me parece verlo por la lista, pero hola. Entonces, sí, y a ti también está aquí. Entonces, solamente deciros que el estudio es, como ya sabéis, porque yo creo que los 3 que habláis español sois los 3 que ya habéis pasado por el estudio, pero es un estudio que realmente nos aporta muchísima información sobre el estado del corazón del niño.
Gracias, Georgia. Si hay otra pregunta de las familias españolas también a Gisele, podemos traducir todos. Eso ahorita es una discusión y cualquier persona puede hacer preguntas o a Gisele o a Georgia para averiguar lo que estamos haciendo ahorita para alcanzar a encontrar una terapia por la enfermedad. Okay? Any other questions that anybody out there has for Georgia, Dr. Brugada, or Dr. Bonne, we'd be happy to answer them.
I have a question. Lindsay from the UK. I have a question, probably for Gisele Bonne. There's been some information in the press recently about a technique, I believe is it CRISPR? CRISPR, and I just wondered whether that was a technique that you are exploring in your research or any of your colleagues are.
Yeah, so this new technique that is called CRISPR, we have discussed this option during our meeting, and there is an ongoing project with one of our colleagues to try to use these techniques first to create some cellular model that would reproduce some frequent mutation and to explore their mechanism. This is the first option. And the second project that has been discussed is to try to remove the mutation from the cells using this technique as well. So these are the two projects that has been discussed with our colleague in Spain, and that will shortly start using these techniques. These techniques is a new way to manipulate the DNA and to try to remove or to add some modification of the DNA. That means removing the mutation or creating a mutation, depending on what we want to do. Either create a model to explore the mechanism, or to try to make a cure and to test a cure by removing the mutation. These are very, very new techniques, and we need first to test this in some cell model in culture to know whether it works.
In Spanish. So, la señora Lindsay de Inglaterra ha hecho una pregunta sobre la tecnología de usar el CRISPR y si eso ya es un proyecto, y es un proyecto que está fundado por la Fundación de Andrés Marcio con un investigador en España que se llama Fernando de Miguel, y él va a usar esta tecnología para hacer dos cosas. Primero, de tratar de introducir las mutaciones más frecuentes de LMNA en una célula, tratar de corregir unas mutaciones que vienen de pacientes en las líneas de células para ver si con eso alcanza a corregir la mutación en las células. Gisele, I would like to ask you a follow-up question on CRISPR because I think this is a question that has come up quite frequently. And
Hello? Hello?
Hello, Juan. We lose you.
This is a question about whether CRISPR...
Could you repeat the beginning of the question because we lose you?
Yes. Okay. My question is whether CRISPR has ever been used to date to cure a human disease. In other words, has it actually ever been used in humans, or is this still a strategy only used in research labs?
So, to my knowledge, this new technology of CRISPR has not been used so far in human diseases, in patients, I mean. It's still at the level of research in the laboratory to test whether it's working, and it seems to be quite efficient in a laboratory, but there is still some concern about so-called off-target, that we want to remove the mutation of the patient, of course, but sometimes this technology can make some effect elsewhere that are not expected. So there is still some concern about the control of side effects of this technology. So it's the reason why it's still at the level of the research laboratory. But several teams work on these new techniques. Try to put it forward towards clinical usage. But there is still some time before it will go to the clinics.
So, para decirlo en español, yo he preguntado si han usado esta tecnología de CRISPR ya en el ser humano, y eso todavía más es una tecnología que están usando en el laboratorio, pero todavía no lo han usado en un ser humano. Y hay algunas dudas sobre los efectos potenciales negativos que, como está corrigiendo la ADN, que es posible que puede corregir un lugar donde no debería corregirlo. Eso todavía es una tecnología muy nueva, pero gracias a la Fundación Andrés Marcio lo vamos a ver si hay un valor de usar esa tecnología.
And we are losing you.
Las líneas de células de pacientes. Okay, I'm going to call in through Skype. Sorry, a little technologically flawed here. I can't find my cell phone. Anybody else have any questions? Joanne, did I hear that you had a question as well? Gisele, could you give us an update on the gene therapy work that you are currently working on?
Yeah, I can briefly do so. So what we have achieved since 2 years, thanks to the support of the Foundation Andres Marcio and L-CMD, is that we have tried to use so-called transplicing technology to get rid of the mutation in cells and mouse models that we have in the lab, and that mimic the laminopathies. So what we have done is we have used this technique that allowed to modify the molecules that produce the mutant protein in patient, and we have designed a molecule that could modify this mutant product and make it to make it as a normal product. So we have designed several molecules, test them first in cell culture, in normal cell culture, then in cell culture from our mutant mice, and we have identified a few molecules that are working and are efficient to transform the mutant product in a normal product. And then, having this very interesting data in cell culture, we have decided to use AAV virus vector to inject this molecule in our mice and to see whether it was also efficient in animal model. So we have done this in normal mice, and this molecule was also able to be present in the mice, and that was very interesting for us. And what we are currently doing is to inject this virus with these molecules that are able to correct the mutant molecule into a normal product in our mutant mice, and those experiments are in progress. So I don't have yet any data to say, but we expect to have it working at least once, and we are initiating a new project to improve this process using oligonucleotide to have a better efficacy in this transformation of mutant product in a normal product.
Okay, so voy a tratar de hacer un resumen en español. So,
Sorry.
Hay dos problemas en el LMNA. El problema es que uno, se necesita corregir el gen, y segundo, se necesita bajar el nivel de la proteína que está mutada. Y en el proyecto que se acaba de cumplir de Gisele Bonne, ella ha usado un método que se llama y para corregir el gen ha usado un vector viral, y ella está en el proceso, o está ahorita inyectando el vector viral y también el transplicing en las ratas para ver si alcanza a ver si la proteína mutada ya está bajada y que el gen está normal. Nomás lo han hecho con una rata y todavía les falta unos experimentos. El problema con el transplicing es la tecnología para bajar la cantidad de la proteína mutada, que el transplicing no es muy eficaz. Ahorita en el proyecto nuevo, ella va a usar una tecnología que se llama oligonucleotides, que están usando en los ensayos clínicos de Duchenne, y va a ver si con eso tiene más eficacidad y puede mostrar que hay una terapia que ha éxito en corregir una mutación en una rata con el LMNA.
Hello, can you hear me?
Yeah.
Yeah.
This is Kyle from the United States. So, best case scenario, if all of this research continues to go well, what's the timeline before this starts getting utilized or potentially used by a member of the LMNA community?
That's a great question, Kyle. I think the first step is there are things that have to happen in parallel. So first, we actually need to know what it is that we are trying to treat. Are we trying to treat the heart, or are we trying to treat the skeletal muscle in a clinical trial? And then second, depending on what we are trying to treat, we have to know what endpoints are the endpoints that we can try to change in a human being so that when we give an intervention or use a drug, we can actually measure either an improvement or no improvement. So that's one step that needs to happen, and that's in part why this LMNA heart study is so important, because it will actually, for the first time, show us over time how the heart changes in children and young adults with LMNA, so that if we were to plan a clinical trial that specifically targets the heart, we would actually know what we can measure and how quickly it changes and whether those changes are related to where the mutation is. It is my own feeling that it is more likely that we will get into a clinical trial. Though some of the drugs that Dr. Warman has been using have shown an improvement at both the level of the heart and the skeletal muscle. So I think probably the earliest that one could see going into a clinical trial would be in 2 years' time, 3 years' time, with a compound that targets the heart based on the data that would then be acquired through the LMNA heart study. Let me just repeat that in Spanish, and then I would be happy to have Dr. Brugada and Dr. Bonne also interject. I think also one other thing, one quick point, is that gene therapy is probably going to take a while, whereas going in with a drug compound that is already being used to treat cancer will probably be an easier bar to achieve. So, en español, Kyle de los Estados Unidos ha preguntado si toda la ciencia se avanza en una manera adecuada, cuál es lo más rápido que vamos a llegar a un ensayo clínico y que podemos tener una droga o una intervención por las personas con LMNA. Y yo he dicho que tenemos que hacer dos cosas en paralelo. Primero, tenemos que estar bien seguros si vamos a ver un cambio, si queremos cambiar un problema en el corazón o un problema en los músculos de los brazos y las piernas. Y me parece que va a ser más fácil de irnos con una terapia al corazón, pero tenemos que saber cuáles medidas vamos a usar para ver si hay un cambio con una droga o una intervención. Y esas medidas vamos a tener por la gran esfuerzo que están haciendo en el ensayo del corazón, porque tenemos que saber cuándo empiezan los problemas del corazón, para entrar en que podemos tener esas medidas. Eso es el motivo porque este estudio prospectivo en el corazón es tan importante. Y una terapia que va a tratar de cambiar el gen, eso va a tardar mucho más tiempo que una terapia que usa una droga que ya están usando en oncología. Y por eso pienso que lo más rápido que podemos esperar, que tenemos un ensayo, son en 2 o 3 años en que quizás podemos probar con la ayuda del ensayo cardíaco y los estudios que está haciendo el Dr. Warman y Dr. Muscular,
aquí vamos a tener un ensayo con una intervención por el corazón. So, Dr. Brugada and Dr. Bonne, I would love to get your feedback as well to the community to answer Kyle's question about whether, how quickly you could
So if I may start, I think, Anne, you already more or less well explained what would be the option we have in front of us and the timeline that would be the most optimistic one to be probably able to go to clinic within 3 years with some pharmacological drug after the development of Dr. Warman and Mushir using proteinase kinase inhibitors. But all this depends on how we will be efficient in going through the path toward clinics with this molecule, because so far there is still a lot of work in front of us to validate the safety and the effect of those molecules and discuss with the regulatory authority to go forward toward clinical trial. But from my point of view, pharmacological drug targeting the heart will be the first to go. As you said, for gene therapy, this will take more time because we are still at the proof of concept in cells and mouse model, and there is still a lot of hurdles to go through with viral vectors, although we use all the knowledge that are currently developed by the community in Duchenne muscular dystrophy. And this is very important for the whole community to have all these advances in Duchenne muscular dystrophy with gene therapy with viral vector. This will help for other indication, other muscular dystrophy who will come, which will come behind.
I would like, have you finished, Anne?
Yeah. You can
okay.
continue, Georgia, sorry.
Okay. No, it's okay. What I would like to add, and it's very important, is that sometimes when we are in science, we try to know improvement, and sometimes it depends on what we measure, we can see improvement, and it depends on what exactly what is the beginning point, let's say, okay? So, in fact, and what we see doing the cardiac study is that if we try to measure improvement once we have already reached a point of no return, probably any type of treatment will fail. Probably if we can predict and we go backwards and we go to a more earlier point of the disease, and that's what we try to see when the disease starts in an early phase, and we put a treatment by that moment, probably we will be able to see an improvement. So the good thing and I think the best thing of working together as a unit or as a whole group of people in different fields like Gisele Bonne is doing and ourselves in the cardiac study is that we need to find when is the best moment to do that, not to reach a point that we cannot go back to even if the drug is good enough. And sometimes it's, should we go before everything is so bad that there's nothing to do about that?
¿Quieres decirlo en español?
Sí, por supuesto. El caso es que en medicina en general, cuando se prueba un fármaco o cualquier tratamiento, lo que tenemos que decidir es qué es lo que vamos a medir. Es decir, si nosotros medimos cosas que ya están muy deterioradas, difícilmente, aunque ese tratamiento sea adecuado, difícilmente vamos a ver una mejora. Si nosotros conseguimos determinar antes de que las cosas ya estén en una situación sin marcha atrás y podemos actuar antes, cuando está empezando el daño donde sea, en este caso en el corazón, nosotros vamos a poder ser capaces, si realmente este tratamiento es eficaz, vamos a ser capaces de ver una diferencia entre el antes y el después. Por eso la importancia de trabajar juntos, tanto la gente que trabaja en elementos tan básicos y trabajando desde las células o desde los ratones, ¿no?, a los que estamos ya trabajando y estudiando a los pacientes. ¿Por qué? Porque es todo este conocimiento junto el que da más sentido a todo lo que hacemos y vamos a intentar encontrar el momento perfecto o el momento ideal para implementar todos estos tratamientos que se están haciendo desde la investigación básica, para entendernos.
Great. Any other questions?
So,
hello, can you hear me?
So is our best, sorry?
Yeah.
Hello?
I think there are 3 questions. I heard a woman's voice first, and then I heard, so is our best step. Why don't we finish the questions that have, to follow up on Kyle's question, and then we'll go to, I think there were 2 female voices as well. Kyle, were you just going to follow up on your original question?
Yep, just wanted to ask if our best, the best thing that we can do as a community then is to raise funds to continue to support this research so that we follow it through and see it through to the very end.
So yeah, so the question is, the answer is yes. You know, even though gene therapy is not around the corner, not here tomorrow, there is a really important point that has not yet been mentioned about why we should continue to support a gene therapy approach. And the reason for this is that the pharmaceutical companies are always going to try and say, what is the best ultimate cure you could give a disease? And that would, of course, be gene therapy to correct the problem genetically. And if we can show that that strategy actually works in the mouse model, then any other drug that we give will probably have less of an effect than a total correction. But we want to know that a total correction is in fact possible. And that will then bring on board more pharmaceutical investment, because then we know what the bar is for how much we can actually correct this disease. Hopefully I've explained that in a way that makes sense. But that's actually a very important point. And of course, if a gene therapy solution is possible, that is in fact the ultimate treatment for any genetic disease. So I'm just going to say that in Spanish. La otra pregunta fue si es necesario la única cosa que la comunidad puede hacer, o una de las cosas es que puede seguir a recaudar fondos para apoyar a la ciencia y los proyectos que ya existen. Y estaba explicando que los proyectos de terapia del gen son muy importantes porque por final las empresas farmacéuticas van a mirar cuál es la cosa más efectiva que se puede hacer por una enfermedad genética, y eso es de curar la enfermedad con una terapia genética. Y si podemos ver que uno tiene éxito en los ratones con esa terapia, eso nos ayuda a saber que una solución que viene, por ejemplo, con una droga al corazón, no va a ser una corrección tan buena y tan eficaz como una corrección genética, pero puede tener una mejoración en la calidad de vida y la sobrevivencia. Y por eso es muy importante, aunque no pensamos que una terapia genética va a existir mañana, de seguir apoyando a la ciencia para avanzar a entender cuál será el tamaño, si se puede decir, del efecto de una intervención o de una droga o de una terapia genética. So I think there were 2 other questions that were coming in, 2 female voices. Please feel free to go ahead and ask your questions.
Go ahead, Jill. Okay, I'll try to talk. I'm not sure if you can hear me.
We can hear you.
Oh, okay. I also posted it in the chat section, but I'll just try to explain it briefly. I'm just concerned because there seems to be not a lot of understanding about the overlap in symptoms with familial partial lipodystrophy and with Emery-Dreifuss. And I was concerned because the lipodystrophy patients are being sent to a different registry when they have overlapping symptoms that might be very important to consider in comparison with Emery-Dreifuss patients. And I was seen in Texas by Dr. Gar. He's basically the world expert on lipodystrophy. And I was definitely diagnosed with familial partial lipodystrophy, so I have both. And I was very shocked to hear that he's inviting patients to come and be seen by him, but he has never had a neurologist refer someone with muscle disease to his clinic in Dallas, Texas. So I'm not sure, can you maybe explain why these two conditions are separated in terms of collecting information when they have so much overlap between them?
The truth and the truthful answer to that question is that we are still trying to understand the metabolic defect. So when we talk about lipodystrophy, for people who are not aware of what this means a abnormality in the body in terms of where fat is distributed and the degree of fatty tissue that one makes. And Gisele can probably give a good scientific explanation in a moment. We definitely see this in all forms of the skeletal muscle disease that is caused by LMNA. So both LMNA-CMD, as you were saying, as well Emery-Dreifuss. You can see patients or people who have very little fat or have fat redistribution. And just like most things in medicine, people who are trained in metabolics are looking at the metabolic issue, and people who are trained in neuromuscular are looking at the skeletal muscle issue, but there hasn't been much crosstalk. And Jill, I think this is a wonderful opportunity, because I know several families on the phone have asked, why isn't there more information on nutrition and how to actually combat the challenges in some individuals in gaining weight. And I think this is a really great opportunity to prioritize this as a potential additional area for research. And maybe one of the next best steps would be to invite this physician from Dallas, Texas, to join us at one of the upcoming scientific meetings. So let me just explain that really quickly in Spanish, and then I would love for Gisele to provide us with an update in terms of what is known on lipodystrophy and the metabolic defect in LMNA. So, en español, la señora ha preguntado por qué les parece que hay un registro que es por los síntomas de la lipodistrofía. Hay unas personas con mutaciones en el LMNA que tienen un problema más con la grasa en el cuerpo y dónde está localizado y la pausidad de la grasa. Y esa señora tiene Emery-Dreifuss, pero también tiene un problema con la lipodistrofía. Ella está viendo a un doctor que se encuentra en Texas, y él tiene su registro propio de personas con lipodistrofía, pero ella está diciendo porque no se juntan toda esa información, porque quizás sería una gran importancia de aprender más sobre el defecto metabólico y el efecto en los músculos. Gisele, do you have any thoughts about lipodystrophy and LMNA?
Yeah. So just to, hi Jill, I was very happy to hear your voice. We know each other for quite a long time.
Yeah,
so
long time.
To answer you, at least in France, we are not doing this separately. We know we are all the community working on laminopathies talk to each other. And what we have initiated in France, and hopefully we will be able to spread this worldwide afterwards, is to set up a patient registry where we collect prospectively all the clinical items of patients carrying a lamine mutation, whatever is the phenotype at first. Congenital form, Emery-Dreifuss, lipodystrophy, neuropathy, or whatever. So this tool has been set up, and in France now we have registered 90 patients. We are currently discussing opening this registry in other languages to to other countries in Europe, first with Italy, because there is also a strong network in Italy. And we are, of course, ready once we will see this registry and the data that we collect are meaningful. We will propose to open it to other countries. This is to make a kind of natural history of any type of lamine at large. What we have initiated with the Andres Marcio Foundation and L-CMD, and it is what Anne highlighted at the very beginning of her talk, we are doing for the early onset type of laminopathy with an onset before 5 years of age, retrospective studies to have the clinical detail of onset of breath difficulties and heart defect. Of course, having nutrition data as well and to have more data about the metabolic aspect is, of course, very important as well. So, to answer you, we know there is overlap. This has been published in the past. We have published this 10 years ago. We know that some patients have true overlap between hemodryphus and lipodystrophy. We don't know yet, we don't understand why some patients have this type of overlap and why some others do not have this overlap. So this is by putting together all the registry that it will be working and have this data together. I know that there is a lipodystrophy registry open for all type of lipodystrophy. You know, registry and databases are multiplying at the present days, and this is just a matter to be connected to each other. And I can reassure you, in the laminopathy field, the experts are connected to each other, and we are willing to share data and understand the meaning of this overlap. Perhaps it's not clear from outside, but I can trust me, it's ongoing.
And Gisele, the UMND LMNA database in France is physician-driven, correct?
Yeah.
Yeah. So, Jill, that's the difference from the CMDIR and the LMNA UMD database in France. So the CMDIR is patient-driven, meaning families register. I think it would make a lot of sense. I know I had said that I was going to reach out to you, I haven't done so yet, but let us set up a call, and maybe I can reach out to the person in Dallas, Texas as well, because it would be wonderful to encourage that physician to refer all patients who also have skeletal muscle involvement or have an LMNA mutation to register in the CMDIR, and that way we can start to share data, and I think this would be very helpful.
If you wish, Anne,
yeah.
If you wish, Anne, what we can propose is to include in the CMDIR some lipodystrophic features that or questions that you could add in your questionnaire from the registry driven by patient.
I think that would be great, because right now I think one of the big problems is that we don't actually know what to measure and how to describe it and how to follow it over time. And I think this is one of the problematic areas when we want to learn more about the nutritional status and the overall metabolic health. I think this is an area that we could use some help in. So I think that would be a great idea, Gisele.
We will discuss
Rachel,
with our endocrinologist and Susanna to provide a few meaningful questions that could be added to the questionnaire.
That sounds great. Let me just say that in Spanish. So, lo que Gisele ha dicho es que por lo menos en Francia, en el database que tiene ella, en el base de datos, que el registro francés, que es un registro que está los médicos tienen que meter la información. Es diferente del CMDIR, donde los pacientes y las familias meten la información. Pero en este registro por médicos, por los pacientes de LMNA, ellos ahorita están metiendo los pacientes que tienen LMNA-CMD, Emery-Dreifuss, girdle 1B, igual como los que tienen una dilatación del corazón o que tienen una lipodistrofía. Lo que está proponiendo Gisele es que ella va a hablar con el endocrinólogo y también yo voy a meterme en contacto con el endocrinólogo en Texas para ver cuáles preguntas podemos meter en el CMDIR para seguir personas con el LMNA y ver si hay cambios en la distribución, lo siento, de grasa o evidencia de una lipodistrofía, y que eso nos va a ayudar quizás a saber un poco mejor si la lipodistrofía también es un problema por personas con LMNA, con CMD o con Emery-Dreifuss. I see there's another question from Joe O'Brien regarding the natural history data, when it will be made available to the community, and whether it will obtain information about the use of steroids. Gisele, did you want to talk about when you think the retrospective data might be made available to the community in a published form, and whether there will be information about the use of steroids?
Okay, so as Anne said, we have collected a total of 168 clinical form from a patient from these retrospective studies, and our plan now is to make meaningful outcome of all the data we collected, and this is a plan for the coming years. We hope to be able to submit and publish it within the coming years. This is for the timeline, because we have so many data, and we really want to extract out of the data very meaningful and important messages for the community. So we want to do it to make a very good and unbiased statistical analysis out of this, and this might take a bit of time, but our objective is to get it published within the coming years, and the fastest will be the better, but we want to make it in the best way. This is for the publication of the retrospective data. And what was the other question? There was
a question about the use of steroids.
Oh, yeah. So this is also an important question, and we have just launched a questionnaire through the physician who sent us the data of their patient for these retrospective studies. And there is an ongoing project, again, for these coming years, to make also a retrospective publication on this specific question of steroid treatment, as it came out that a few of the patients has already been treated on steroid, and the outcome is not always identical, so we want to be also very careful and make it properly. So we will evaluate the retrospective data in detail for those patients who got some treatment already, and we are designing currently with Susanna Quirano-Roy in Garches Hospital in France a prospective study for those patients who will be put on steroid, to evaluate whether it has a positive effect or not.
Gisele ha dicho que ella piensa que dentro de un año van a tener una publicación de los datos de la historia retrospectiva, y también que Susana Quijano-Roy, una neuróloga en París, va a empezar un estudio prospectivo para evaluar la eficacia de usar esteroides en LMNA. Great. Dr. Brugada, can you also update us on when you think data might be available from the cardiac study? Because as you've pointed out, the cardiac study or the heart study is going to optimize care for people with LMNA from a heart standpoint, and may actually change how care is provided.
Yeah, for sure. We are probably in the following months, probably from the following 6 months, we'll have recruited enough patients to start doing the analysis of the data. But of course, what we see, it's not something immediate. What we will see is the following up of these patients, that we will receive all this information from the subcutaneous devices that we will have implanted, and probably I think in let's say a couple of years from the starting of the study, we will have some data to share probably. So but we already know that at the first month after we started the study, we had 2 big events that made us speed up the inclusion of patients in the study. I just posted my email on the chat, so you can easily share or email me if you have any questions on the cardiac study. And I think it's easy if you have certain questions that you would like to know more details about.
Great. Are there any other questions?
I have a question about the pharmacology aspect.
You know, can you speak up just a little bit? You're a little bit quiet.
Sure. Sorry about that. I have a question regarding the possible drug therapies. I'm Deborah. I had a heart transplant 7 years ago, and I've been on sirolimus for 6 years. I believe sirolimus is one of the drugs that's being investigated to help with our disease symptoms. I'm just wondering if that is being studied as it affects cardiac muscle, or is that being studied as it affects both skeletal and cardiac muscle?
Great, thank you for your question. I could barely hear you, but I think what you said is that you had a heart transplant and you've been on a AKT mTOR is how they are called, drug, one of the, like a temsirolimus, or maybe you've been on everolimus, is that what you said?
That's correct.
Okay, so you've been on everolimus for the last 6 years since your heart transplant. And the question was whether that particular type of a drug is being evaluated for an effect both on the heart and the skeletal muscle, or just on the heart. Is that the question?
Yes, that is. Thank you.
Okay, great. Perfect. So the only data we currently have so far is from Dr. Warman's lab, and it was data that was done together with Dr. Antoine Muchir. And that was a very short-term study where they used temsirolimus to evaluate an effect on the heart. What is really needed is a head-to-head comparison in the mouse model of a drug like temsirolimus or everolimus versus the protein kinase inhibitor, which is a different drug compound, to see whether one or the other impacts the heart better and survival better. One of the confounding factors here, at least in the mouse model, is that we don't really know what the mice are dying of. So it is possible that you could improve the heart function, but that the mouse still dies because it is actually not dying of the heart issue. And that makes it a little bit challenging to know, when you're doing a head-to-head comparison of the two drugs, what is actually really kind of, what are outcomes you want to compare, and whether you're really comparing two drugs effectively in terms of what effect they might have in humans. But it's very interesting to hear that you've been on everolimus, and I'd like to just ask you whether you'd be willing to share whether you've noticed any improvement in your muscle strength.
Well, I'm actually on sirolimus rather than everolimus. That came out a little after I was put on the first one. But in my opinion, I haven't had, my muscle strength hasn't worsened very much over the last 6 years, but I'm still weaker than I was before starting to take it. So I just think it's a matter of being 6 years out further into the disease.
Yeah, and those are some of the challenges are, you know, if a drug slows disease progression but doesn't necessarily improve you to a prior status, that might in and of itself be an effect size that we would all want to see. But it's hard to measure that without knowing, you know, what the natural history of the disease would be in somebody who has had a transplant with your mutation. Do you see what I'm trying to say? So this is actually one of the projects that I proposed to Dr. Warman, is to do a head-to-head comparison of a drug like temsirolimus or everolimus versus the MAP kinase inhibitor in order to get more information on which drugs, or potentially in combination, whether you see an even greater improvement. And that's a project that I've asked him to put a proposal together for that would require additional funding, but I think it's an important project given that the cardiologic community is also very familiar, or more familiar I should say, because of heart transplants and the use of these type of compounds that came from the cancer world in neurofibromatosis, where you may also have some lesions in the heart. They are more familiar with the use of those compounds as opposed to MAP kinase inhibitors. And then the final thing that I have to add for all of these drugs is that there potentially is also a negative impact on the heart. And so you have to really understand, and this is what Gisele was saying before, in terms of additional studies that are needed, it'll be important to sort out how much of the drug is giving a benefit effect versus if there's an off-target negative effect as well. And who does it make sense to give this drug to chronically with this condition. So I'm just going to stop there and explain everything in Spanish real quickly. So la señora acaba de decir que ella ha recibido un trasplante de corazón hace unos 6 años y que tiene una mutación en el LMNA y que desde ese punto, porque tiene que tomar una medicina para prevenir la rejección del trasplante, está tomando una medicina que más o menos es como temsirolimus, que es la droga que Howard Warman ha estudiado con Anton Munchir en la rata. Y ella está tomando este medicamento desde hace unos 6 años y ella ha notado que está un poco más débil, pero no sé si la debilidad en los músculos. Su pregunta era si la droga, si lo han estudiado en la rata, si la droga está más empeorando o ameliorando el problema del corazón o de los músculos. Yo le he explicado que en el estudio de Howard Warman fue un estudio bien corto, nomás por 2 semanas, para ver si hay un efecto por el corazón, pero no por los efectos a los músculos. Y yo les he preguntado a la señora si ella ha notado si sus músculos están más débil o más o menos van por lo mismo o están mejor desde que ha empezado esa medicina hace unos 6 años. Y ella ha dicho que los músculos están un poco más débiles, pero ella no sabe si eso nomás es el desarrollo de la enfermedad o si sería peor si no hubiera tomado esa medicina, que está una medicina usada para prevenir que el trasplante está, no sé cómo decir, rejected. Georgia, ¿cómo dice eso?
Rechazado.
Rechazado, gracias. So, y yo les he explicado que he hablado con el Howard Warman porque pienso que todavía necesitamos un proyecto más, y el proyecto sería un proyecto en la rata de comparar una medicina como el temsirolimus o el everolimus, que es una medicina que los cardiólogos ya están más o menos, saben cómo usarlo, como lo usa por los trasplantes y también se usa en la neurofibromatosis cuando tienen unos tumores en el corazón, y compararlo a la droga que Howard quiere estudiar ahorita, que es un MAP kinase inhibitor, para ver cuál de los dos tiene el efecto más grande, positivo en la sobrevivencia, igual en los músculos como en el corazón. So, yo he pedido a Howard que hace un proposal que nos da una idea de un proyecto que hace exactamente eso y porque pienso que es muy importante para nosotros en la comunidad de entender un poco mejor cuando uno compara las dos medicinas, las dos drogas, cuál sería lo mejor por la LMNA en la rata. Siempre se tiene que calificar que quizás no va a ser parecido en el ser humano, pero en la rata, ¿cuál sería mejor? Great. So the meeting, our meeting today was supposed to go for an hour, and I'm sure there are additional questions. And I know that some people do need to leave. I know that Dr. Brugada needs to leave right now. Are there any other questions for Dr. Brugada? And then maybe we can put you in touch with her directly if you have specific questions for Dr. Brugada. So just a quick question, any questions for Dr. Brugada? So.
Thank you, Dr.
No.
Brugada.
Thank you. I don't know if it's going to last a lot longer, or
I think we'll probably wrap it up in the next 5 minutes, Georgia.
Okay, great. So I can just wait, of course. Yeah.
Okay, fantastic. So I would think we have time for one more question, and I want everybody to know that we hope that this is just the beginning of a discussion and an effort in our community to raise awareness of the science that is going on, of course, to assist us in raising funds to promote additional science and science that is focused on funding treatments and understanding the disease course in humans. So I would like to just ask if there's any other questions. Si hay una pregunta más, tenemos tiempo por una pregunta más. También por las familias españolas, si tienen alguna pregunta. Okay, I think there are no further questions. So first, I would really like to thank our co-funders and collaborators in all this funding, the Andres Marcio Foundation, Jesus Marcio and Beatriz Olona. And then I would like to thank our wonderful colleagues and scientists who have joined us today, Dr. Georgia Brugada and Dr. Gisele Bonne, for their incredible commitment to the LMNA community and the endless hours that they have invested in the community, including being on the phone on a Saturday night to answer questions. I'm sure both of them would be happy to follow up with you individually if you have additional questions. We are going to, we have recorded this webinar, and L-CMD will convert it to a YouTube video, and we'll distribute a link to this video through the registry, and we will post it on the LMNA Facebook, and I know Salome will post it on the Emory-Dreyfus Facebook, and we'll post it on the Facebook and the Andres Marcio Foundation Facebook. And then what we will plan to do is to hold a webinar at least once a year, if not twice a year, to continue to provide the community with updates on the science and on the clinical research, and to answer any questions that you might have. So let me just say that in Spanish. Primero queremos darle gracias a nuestros amigos en España y que son nuestros colaboradores, la Fundación Andrés Marcio, y también queremos decir gracias a los dos cientistas y médicos que están con nosotros hoy, la doctora Brugada y la doctora Bonne, que han hecho una gran esfuerzo y siguen haciendo una gran esfuerzo por toda la comunidad, especialmente de estar en el teléfono a sábado por una hora por la noche o por la tarde, y les queremos agradecer. Y esperemos que eso nomás es el principio de una discusión entre toda la comunidad, y que seguimos con unos webinars o 1 o 2 veces al año para discutir cuáles son las noticias de la ciencia y de los proyectos, y para decirles también en qué nos podemos ayudar en recaudar fondos para apoyar todos estos proyectos. With that, I would like to say a good morning, good afternoon, and a good night wherever you are in the world. And we will be in touch shortly with the YouTube link for the recording of this webinar. And I think we're going to call it a day. Thanks so much for joining us.
Thank you, Anne. And I would like to add here that we are talking to Gisele Bonne, and after all the big terrorist attempt that Paris have suffered last night, and I think we should be really thankful to Gisele to share a so difficult day in France today. So thanks, Gisele, and we really feel very bad about all the situation that you are living.
Many thanks, Georgia. Indeed, it's a very awful day today for us, but also for the whole world, because it's so awful we cannot even imagine that it had happened. So I had received all the day long messages from all my colleagues throughout the world, and I really appreciate all the support you provide us. But I think the whole world has been attacked yesterday, and I think, well, having this type of discussion today makes me thinking that there is better things to do than killing people in the world.
Oh yeah, for sure.
Thanks a lot for your support, and I wish you all the best. And it was a great honor for me to participate today to this conference call. Thanks a lot.
Thank you so much, Gisele.
Thank you, Gisele.
So bye-bye, everybody.
Thank you so much. Goodbye to everybody.
Thank you, everybody. Goodbye.